Hello everyone, this is Fredrik Vermling again. In this video we're going to talk about how to analyze CRISPR screens using gProfiler, another really nice tool for analyzing gene sets. This is gProfiler, how it looks, and you paste your genes here. And you choose your organism that you want to work on, and this has a lot of them. So it's really open for a lot of things, but in this case our genes are Homo sapiens. Usually by default these highlight driver terms in Go are chosen, so let's click that one and then you press run query. You can also click here to get sort of suggested gene sets. And what comes up then are sort of a selection or suggestion about different gene sets, Go terms, KEGG, reactome, etc. That comes up and are significant. If we go into detailed results, we can look at them in more detail as the name implies, and then we can get sort of here our Go term molecular function. We get the Go term name. Note here you can press it, and then you can get up sort of which genes that are involved in it. You can export it. So if you want to have these Go terms, actually this is a way, if you want to know which genes are part of the Go term, you can go to this gene convert and paste it there and easily get out the genes, for example. You get then some sort of a statistical test here showing the link to your gene set, and also interestingly you can see sort of which genes that are linked to what in here, to the different ones. And if you have a lot of genes, this is not going to make sense, but when you have fewer, it sort of makes you can look at it visually like this. Different colors explained here, but it's essentially, as I understand it, coming from different data sources. You can press stats here, and then you'll see that we queried 51 genes. This gene set the top one has 47 genes, and there were 5 of them that were overlapping. So you can look at them like this, 1, 2, 3, 4, 5. Or you can also press the number here, and again they come up, and if you want to work with them in some other way. So you have Go terms, different ones, and again if you pick these highlight driver terms, it says some most relevant terms in gene ontology are highlighted. So you can if you want sort of focus more on these blue ones, for example. So we have Go terms, KEGG, reactome, wiki pathways. We get transcription factors, microRNAs, etc., etc. Right? I want to show one more thing about this, which is that you can say show only selected, and then you see only the ones that were selected. Now we only saw the ones that were selected, and they were selected because they were driver terms. So we can remove that if we want to, and then we can sort of select the ones that we are interested in, show only selected, and then you have a much more condensed image here, right? And then you can for example export that as a CSV file, and you get data here on sort of what's in it and which genes are there. So you can get interesting information, or some files that you can keep for. And you can also make images, say these sort of images. In a way, this overlaps a bit with several others of these tools that we're talking about in this series. It sort of gives you Go terms and etc. I do like the way that sort of genes line up like this, especially when it looks something like this. So if you look at the KEGG here, and if you look at the different terms that are identified here, it could give the impression that there is in our gene set there is or data set here there is a very wide heterogeneity or diversity in what it's linked to. But I think it becomes graphically very obvious when you look at the genes set up like this that no, this is not representing something very diverse. It's actually more or less the same genes. So the diversity here is that, or sort of there's a lot of this tells us that there's a lot of these terms that are sort of overlapping in their gene sets, and I think that's something most people know. But sometimes when you look at this, you could see small cell lung cancer and measles. That's very diverse, but actually it's just it's the same genes in this case. I think that could be pretty useful. Another thing I want to highlight here is query info, and this is a topic I'll probably come back to more, because our input was 55 genes, and you can see that 52 of them are included here, while 3 of them actually were not, and that's because these gene names were not recognized by this tool. And this is a very common problem when you start working with gene names and from different sources, that there are different names are used essentially. And again, this is something everyone, that's when you start working with more with these sort of things, you need to sort of address and think about, because theoretically it could be a super important pathway here that you miss because the tool didn't understand which gene you were talking about. And here in gProfiler you can press the gene and then you can see sort of it's relating, okay, the VPAL, which is what our input was. It might also actually be spelled something like this, VAPL, right? Human gene, VAPL. And we can test that, sort of putting it back, VAPL, and then rerun it. And then now we'll see we inputted the 56 genes, but since VPAL VAPL didn't show up here, not included, then it means that this other gene name was identified by gProfiler. And then of course you need to think a little bit about, was this correct? Did I make the correct sort of change here? But this is something that could be good to look into, because it could bias your data. I also want to just mention this gConverter. I talked a little bit about it already, but I think it could be useful. One thing is, by just pressing, putting your genes in here, you can get things like, so in this case, ensemble genes, right? But there's a lot of other sort of outputs that could be coming from this. So you could convert your gene name into an alias that could be relevant for another software, for example. And you can export these and sort of use them in other ways, right? So here is med12 is the gene name, and this is the ensemble gene name, right? So I think that could be useful. And it could also be useful, and this is also not uncommon, that you might sort of work with a mouse set of genes, but you might be interested in looking for human genes and reversed here, right? So here you can try to look, you have human genes and you want to identify sort of the mouse genes, and then you get a list, and most of the time when you go from human to mouse or the other way around, it's just changing from capital to non-capital letters, right? Lower cases. But there's certain genes that have different names, like TP53 in humans is TRP53 in mouse, right? So this is very common. Here CIS also seems to have 2 different names potentially in mouse, and you need to think about if that's relevant or not. And you can find some of these that are not found. It's not translated well. So I think that could be this definitely has applications. It could be useful sometimes when working with sort of gene sets and trying to understand sort of connections between them. All right, so this was gProfiler, also another useful tool for analyzing CRISPR screens. I hope you learned something. I hope you watch the rest of the videos in this series, and I hope I see you again. Bye.